All right. So my name is Merrill Asp. I'm in the physics department at Syracuse University, and I'm here to present the work on bacterial biofilms that I did with my advisor, Alison Patteson. So let's just quickly look at what we know about biofilms. As a physical system, there's a lot going on in biofilm growth. So you can see in the video, as bacteria encounter a substrate, they have a tendency to colonize it, to spread outwards. And that outwards growth is driven by active fluid flow. As you can see in this sketch, the cross-section, the bacteria are producing polymers, exopolysaccharides, or EPS, are producing polymers at the edge of the biofilm in very high concentration that drives fluid up through the substrate they're on by osmotic pressure. And as that fluid is driven up, the biofilm expands. The uptake of that fluid also brings with it the nutrients that the biofilm need to convert into EPS to continue their growth. And we can see all kinds of interesting morphological features in this video. But in particular, we want to understand better what the bacteria can sense about the substrate that they're on. So we can perform experiments changing the substrate to see what inputs affect colony growth. Now, there are some known effects in the literature that are based on substrate. For instance, growing biofilm, different concentrations of agar shows fairly clearly that as you have a higher agar concentration, you tend to have less biofilm growth. You'll have these smaller biofilms, and that's largely been attributed to porosity, that as you have a higher agar concentration, the network is very tight, the pores are small, and it's difficult for the bacteria to drive fluid through the substrate. They're essentially just being choked of nutrients. But there are a lot of other perspectives that we can take for looking at that interaction. In particular, we ask, how does substrate stiffness affect biofilm growth? Increasing agar concentration doesn't just change pore size, it also makes a stiffer substrate. Now, biofilms themselves are quite soft, but we know that they are able to colonize a very wide range of substrate stiffnesses. This leads us to hypothesize that there is some mechanical coupling between the biofilm and the substrate, and that's really what we're interested in investigating. Now, going back to agar, as we increase the agar concentration, not only does the stiffness, the elastic modulus G prime, increase with agar concentration, we also have more viscous dissipation of energy. That's a viscoelastic property of agar. So there's a lot of variables that are changing with agar concentration. If we want to isolate just the effects of stiffness, it would be helpful if we could design a purely elastic substrate, such as a polyacrylamide hydrogel, which, as you can see here, as the stiffness increases, we actually have the same almost zero amount of viscous dissipation, which suppresses the number of variables that we're changing with stiffness. Another advantage of using polyacrylamide hydrogels as your substrate is the fact that the pore size and the stiffness can be independently tuned. And that's because the polyacrylamide hydrogels are formed from a network of acrylamide monomers that are linked with another chemical, bisacrylamide. So you can increase the stiffness of these hydrogels either by giving more monomer or putting more monomer in, or by increasing the amount of crosslinker, which, as you can see in this diagram, increases the stiffness but maintains approximately the same pore size. So that's what we did. We prepared many different polyacrylamide hydrogels on glass cover slips, and we inoculated a bacterial solution of Serratia marcescens with growth medium onto each of those gels. Now, Serratia marcescens produces this bright red characteristic pigment as it grows, which you can see in this overhead view of a fully grown biofilm after about 15 hours of growth. So we were able to use time-lapse microscopy to take a look at individual sections of these colonies as they were growing to produce videos like this, where we can see the actual growth dynamics. So after we ran all of these experiments, some of these videos started looking like this. What we saw, as you can see on the left, we have a softer hydrogel, and on the right, we have a stiffer hydrogel. But there's a noticeable increase in growth on the stiffer substrate, which surprised us. That biofilm gets an edge, gets a lead as a function of that stiffer substrate. And that was surprising. So we went back, we did an agar control, again on low concentration of agar, high concentration of agar, and we saw, or we confirmed what had been in the literature, that on agar, there's less growth with a stiff substrate. So that convinced us that we had seen something novel in the growth experiments on polyacrylamide hydrogels, where we had isolated just the effects of stiffness and where we saw increased growth on stiffness. Now, in order to quantify this, we developed a custom automated boundary detection script in Python. For those of you who are interested, that's available on GitHub for you to look at, which essentially traces the boundary of the biofilm at all these different points in time. And from that information, we can calculate the boundary velocity. So after quantifying these videos, this is what we saw. We saw a conformation numerically between biofilm velocity, the boundary velocity, and stiffness. Now we saw that particularly in this window, this low stiffness window, up to about 6 kilopascals. And that effect seemed to saturate at higher stiffnesses, but particularly those early velocities when the biofilm is just starting to colonize, we see increased velocity with stiffness. So that's suggesting something's going on in this coupling. In order to investigate that, we embedded fluorescent tracer particles in the substrate so that we could see the deformation in real time as the biofilm was growing across that polyacrylamide hydrogel. So you can see the biofilm highlighted in red or without it. And we were able to directly observe these displacements on the orders of tens of microns. If we look at the displacement vectors themselves, what we generally saw is that the biofilm edge does push the substrate somewhat outwards, but within the bulk of the biofilm, there's a little bit of an inwards pull towards the center. Now we're still trying to understand and investigate the nature of this physical coupling, the nature of these forces. But in conclusion, we've been able to see that polyacrylamide hydrogels as a substrate are more tunable than agar and are able to isolate some of these effects of stiffness better than you can on agar substrates. In particular, we were able to see an increase in the rate of growth with substrate stiffness in this particularly soft window and at early times of growth. And we were able to directly observe the shear deformation of the substrate as the biofilm was growing. So I'd like to acknowledge the support that we received from our NSF grant and to thank you for your time.